Hello guys, today we are going to talk about Juxtaglomerular apparatus. Before diving into the details, let me tell you a story. Have you ever wondered how kidney knows that the blood pressure of the body has been lowered? How the kidney knows the glomerular filtration rate has been decreased? How the body knows or the kidney knows or the tubule knows that sodium and chloride concentration has decreased? Who senses that? Who talks to the kidney? Who tells the kidney to do that? The process of absorption or the process of excretion? So that's defined the autoregulatory function. The circulating concentration of the electrolytes, the body volume, that means in terms of fluid and electrolyte volume, who senses that? Who is the structure who is telling the kidney to regulate these things? Is there is involvement of brain? Is there is involvement of the spinal cord? Is there is involvement of any other visceral structure, or it is only the kidney who is playing the important role? So yes, that's known as the juxtaglomerular apparatus. When I am saying juxta, that means it is near, that is the junction. That's defined as the juxta. Glomerular, that means these are near to the glomerulus. We have seen in my previous lecture that glomerulus is a dynamic filtrating unit. It's not alone. It is having the well-defined basement membrane, the fenestrated capillary epithelium, and the most important, the podocytes. Their interaction makes it a huge filtration plate. That's why, naturally, in the course of evolution, cells, sensors, stimulants, receptors, they are developed all along the length of the glomerulus, so that second to second, minute to minute, precise regulation of fluid and electrolyte volume will be carried out. So with this very introduction, let us see what is known as juxtaglomerular apparatus, how it is organized, how it senses, how it maintains the dynamics inside the body. So before going into the details, do not forget to like and subscribe my channel. So let us begin with this word, juxtaglomerular apparatus. In this particular lecture, we are having three parts. This is the first part where I will introduce juxtaglomerular apparatus. In the second part, we'll draw a diagram where we'll see the cells of the juxtaglomerular apparatus. And in the third part, we'll see the dynamic signaling and sensing mechanism by which this juxtaglomerular apparatus operates. So let me introduce this term juxtaglomerular apparatus. What is this? It's a group of specialized cells present in, around, or near glomerulus or the glomerular capillaries. This is the actual point, that it is a group of cells. This acts as autosensor. That means it senses the salts or the electrolytes. Particularly, I am talking about the sodium and the chloride. It senses the volume. Particularly, I am talking about the fluid volume. That means it senses the degree of hydration of the body. How much of water is present in circulating form. And third, it senses blood pressure. One of the most important functions of kidney. Usually we say that kidneys are the structure which are producing the excretory waste, but we should not forget that the regulation of the blood pressure in a controlled level within a very specified limit is regulated by the kidneys, and in specific juxtaglomerular apparatus. So I told you three functions, that it senses the salts, it senses the hydration, it senses the blood pressure, and all together it acts. Next, what is its outcome? This juxtaglomerular apparatus, it regulates important, fluid volume, electrolyte volume, this is the fun side, electrolyte volume, it regulates blood pressure, it regulates glomerular filtration rate. Now these are the 4 outcomes. These are the 3 sensors. These are the sensing mechanism. This is the outcome. Now coming to the point, who are those who are stimulating? That is reduced sodium, reduced Cl minus, reduced glomerular filtration rate. These are the points which are making it to be sensed and decrease renal blood flow, decrease renal blood pressure, decrease arterial blood pressure. So these are known as the sensor, the dynamic sensors. That means these are the parameters. When they fall from their normal level, then these cells jump into the picture. Next, the last point, who are they actually? So the answer is they are the macula densa. Unless we draw the diagram, we cannot understand. That is macula densa. Who are more? The important, the juxtaglomerular cell. Stay tuned for my next lecture on renin, angiotensin, aldosterone system. There you will see what is the important function of this cell. The third one, the mesangial cells. Oh my God, mesangial cells are again 2 types. One is just between the glomerular capillaries and one in the outside the capillaries between the macula densa, efferent and apparent atoll, the triangular area. That's known as the extraglomerular, mesangial cells. Mesangial cells. Or you can use the word lattice cell. Or there is another name is there, cells of Gourmatic cells. So these are the fundamental cells of this particular structure that's known as juxtaglomerular apparatus. One is lattice cells, second is mesangial cell, third is macula densa, fourth one is this juxtaglomerular cell. So guys, with this very brief introduction, we came to the end. That end is that juxtaglomerular apparatus is nothing but a group of sensory cells which sense the very precisely, a small change in the concentration of the salts or electrolytes, fluid and blood pressure, by virtue of which it will activate its autoregulatory system, and the kidneys will try to regulate the function so that physiology will be maintained. After knowing the cells, let's draw a label diagram where we will see how these cells are present and what functions they carry out. So this is a beautiful diagram which will explain the various cells of the juxtaglomerular apparatus. Dear friends, always remember there are several questions from this. Number 1 point, which is the sensor for sodium and chloride? Answer, macula densa. Who is that cell which secretes renin? Answer, juxtaglomerular cells. Name the cell which secretes prostaglandin in the glomerular bed. Answer, lattice cells as well as mesangial cell. Who secretes the basement membrane of the glomerulus? Answer, mesangial cells. Who regulate the autocontractility around the glomerular capillaries? Answer, mesangial cells. Name the phagocytic cells of the nephron. Answer, mesangial cell. Macula densa stimulates the juxtaglomerular cells. True or false? True. So that means there is a group of questions which are residing in this juxtaglomerular apparatus. That's why I have designed this lecture so that you people can well understand. Who is the stimulant for renin, angiotensin, aldosterone system? Then we say that it is the increased potassium, decreased sodium, then how that is being activated? Answer, macula densa. Where macula densa are present? It is present in the DCT, just near to the efferent and efferent arteriole. Why these are known as macula densa? A mass of cuboidal cells in the DCT who are very much concentrated. Can you believe? There are 300 to 400 cells are present per microsurface of, that means per mm. It means it shows that they are densely packed cells. What they contain and why they sense? They contain sensory nerve endings. Which kind of sensory nerve endings are present? Sympathetic nerve endings. And can you believe there is a very important question comes, who regulates the GFR? Answer is macula densa, answer is juxtaglomerular cells or Lassus cells or mesangial cells. Of course, mesangial cells are contractile nature. Who increases the peripheral resistance or resistance in the afferent arteriole? Who increases the resistance in the efferent arteriole? Altogether, I can say this is a group of questions. So stay tuned and let's see and let's draw the diagram so that we can well understand how this phenomenon is going on. I will make this structure very, very easy so that in one side I can draw a cellular diagram and in the other side I can tell you the functionality. So what I am drawing, just try to understand. The diagram shows a curve-shaped structure. The curve-shaped structure is known as the Bowman's capsule. From here, the proximal convoluted tubule arises with the broad border cuboidal cells. Here, what you are seeing, now you are seeing the renal pole here, the tubular pole or the renal pole from where the PCT arises. And this is the area which I can use the word vascular pole, where the afferent and efferent arteriole enters. So when the afferent arteriole comes, it is wider in diameter. It goes inside. Then it branches to 4 to 5 secondary vessels, which again undergo the process of coiling and twisting, so that 50 number of, 60 number of, 70 number of coiled capillaries will appear depending on the species. And thereafter, this is the narrow in diameter or the efferent arteriole comes out. So this diagram is showing this is the glomerular capillaries which are present in a tuft-like structure. Porous endothelial cells are being present and they are the cells which are surrounding this. Those are known as the podocytes. This is the diagram of the glomerular capillaries and the podocytes. So this much of diagram we have drawn in our previous lecture as well. Now see what's going on. What's the speciality? So the speciality is that some cells are present here just along the capillaries. These are some of the cells which are present along the capillaries, and this is known as mesangial matrix, and the cells are known as the mesangial cells. Here you are seeing some of the cells which are present along the efferent arteriole. These are group of sensory cells, very, very dynamic. Granular Golgi complex makes it more suitable for the purpose of this juxta function as well as granular function. These are the cells. Here only 30% of the cells are present along the efferent arteriole, but if you see here they are so much, and that's the nothing but the efferent arteriole. So in this diagram you are seeing this is the afferent arteriole along the vascular pole where they are entering the Bowman's capsule and they are forming the glomerular tuft. This is what you are seeing, the efferent arteriole, and this is smaller in diameter compared to the efferent arteriole. The blood is passing this side, the blood is coming from this side. So now the cells, the beautiful cells, the mass of cells, what you are seeing here, this is the cell. That's known as it is junction, it is juxta glomerular cells. And these are granular cells which are the major regulators of the juxta glomerular apparatus. What cells you are seeing here, these are known as the mesangial cells, the autocontractile cells of the nephron, the mesangial cells. So this is number 1, this is number 2. And now I have taken the cross section of, I can say I have taken the cross section of the distal convoluted tubule, and here this is the distal convoluted tubule, and these are some of the cells which are cuboidal in appearance, but there are some cells are there which are densely packed. These are those cells. These cells are very important. What you are seeing here, I can say these cells, these are the cells which are known as the macula densa, the densely packed cuboidal cells, macula densa. So this is the number 3 cell in our discussion. And after that, you are seeing some of the cells which are in miniature amount. These are here, and these are the 4th category of cells. This is extraglomerular, outside, extraglomerular mesangial cells. So they are present outside the glomerular capillaries in the triangular area. So I can draw a little bit more cells so that you can understand the juxtaglomerular cells are the predominant cell of the juxtaglomerular apparatus, more towards the afferent arteriole, less towards the efferent arteriole, and they play a very critical role in the detection of that blood pressure and sensing from the macula densa so that they will secrete the renin. So this is the 4th category of cell in our discussion. So if I can say the diagram and the functionality, then let us write their function, one function along their course. So the mesangial cell, it is phagocytic in function, contractile in function because it contains the actin-like microfilaments. This point is important. It secretes the basement membrane. That glomerular basement membrane is synthesized by this. It secretes mesangial matrix, another matrix where these cells are being present. It secretes the prostaglandin. That's why it is contractile in nature, the prostaglandin. That means altogether, these are the important functions of the mesangial cells. Once I'm coming to the extraglomerular mesangial cells, their function is very important. They are, earlier it was not known, but now they are believed to secrete the prostaglandins. They are the lysis cells, or the cells of Gumatry, or the extramesangial cells. Then I'm coming to the most important, that is the macula densa cells. They are the sensors of low sodium. They are the sensors of low chloride, and that's why they act as the potential sensors of electrolytes. And if I come to the juxtaglomerular cells, yes friends, the very important function is the secretion of the renin, which is the key regulator of renin, angiotensin, aldosterone system. And once I am saying this about the renin, that means we have to understand its mechanism. If I can say another important function, what I have told you in blood clotting, that is thromboxane A2. This is also secreted by the macula densa. So 3 functions: when it is stimulated when chloride level is low, it is stimulated when sodium level is low, it secretes thromboxane A2. It also has the stimulation on the juxtaglomerular cells when the BP, the renal blood pressure decreases. Mesangial cells, lot of functions I told, and the most important function, it is supporting to the glomerular capillaries. And of course, the most important cell, which is very rich in endoplasmic reticulum and Golgi complex, which is secreting a glycoprotein hormone renin, which has the activating role on the angiotensinogen, and it is ultimately converted to angiotensin 2. Stay tuned for my next lecture. That's a dynamic lecture on the RAS and how the RAS regulates the functions of blood pressure and the electrolytes in the body. One of the most important systems in the long-term process of regulation of blood pressure. So that will be in my next lecture. So let's draw the final flow chart And that flowchart will show this beautiful sensing how these structures work together. So I am coming to the final part, and this final part means it shows that how beautifully the mechanism is operating inside the body. Because whenever we understand physiology, we must understand the signaling mechanism, because it is very difficult to know that whether such functions occur in the body or not, unless and until you have an idea that yes, these are the functions which are occurring in a minute to minute or second to second base. You must also understand there are some sensing structure. You must also understand somebody is there to tell that it is decreased, it is increased, or we need some case of homeostasis. So that makes these processes of signaling always very important, very significant, and as well as very interesting. So let us draw the final diagram. That's what I was saying, the final flow chart. That how this juxtaglomerular apparatus is important. So decrease arterial pressure, arterial blood pressure or pressure. This is the term which initiates the pathway. This down arrows means decrease. And once this is decreased, there is reduction in glomerular hydrostatic pressure. Already I told in my previous lecture, but in the coming lecture we'll deal about more. This is around 60 mmHg. Then the glomerular hydrostatic pressure decreases. Understand one by one point. Once this is decreased, that means the GFR decreases. This causes the reduction in the glomerular filtration rate, which is around 125 ml per minute. Once the GFR decreases, what happens? There are two possibilities here. One, increase reabsorption of sodium and Cl minus in the loop of Henle, particularly in the ascending loop of Henle. Once there is increase in the reabsorption, then what is the result? The result will be decrease in sodium, decrease in Cl minus to the DCT. That means the DCT will receive less sodium, less Cl. What will happen? That means this is that stimulation of macula densa. So this is the beauty of this signaling. This the macula densa is now stimulated. That's what I was saying. It's the sensing. The sensing is important. And once this sensing is important, now what will happen? That means already the macula densa is sensing, and what it will do. So I can draw one more thing, that is from here also. That means there is stimulation of macula densa. That means macula densa is now stimulated. What it will cause? So from here one arrow, the flow chart goes in this direction, and another arrow will go from this direction. So I have two parts, and both these parts are independent to each other, and this will now cause the stimulation of juxtaglomerular apparatus. This is the beauty of this mechanism. So once this juxtaglomerular apparatus, the juxtaglomerular cells, they are stimulated. The macula densa will send the message. This is through sympathetic route. What will happen? The renin will secrete. What is the target of renin? It will act on, this is my next class, which is in detail. It acts on the angiotensinogen. What will happen? Now ultimately, the master molecule, that is angiotensin 2, is formed. This mechanism is really very dynamic. Now just to see, once this angiotensin 2 is formed, where it acts? It acts on efferent arteriole. See the beauty. It will act on the efferent arteriole, and the efferent arteriole resistance, it has to be increased. So this is the beautiful outcome of this sensing mechanism. And once this efferent arteriole increases, then it causes the glomerular hydrostatic pressure to increase. That means here I can put a negative sign. From here we have started decrease in the glomerular hydrostatic pressure. So now due to this angiotensin 2, what we are doing, now we are showing that there is an increase in the resistance of the efferent arteriole, and thereby this glomerular hydrostatic pressure is now compromised. That means increase. So whatever they written, I have written now they will gradually stop. How beautiful this is. Then what will happen? Now this macula densa also stimulates. See how simultaneously they are working. It increases the efferent arteriole efferent arteriole resistance, and once it is also increased, see how beautifully this also negotiates this glomerular hydrostatic pressure which was decreased. That means it will now increase. So altogether macula densa is playing a role model. Here you can see, here you can see. But what is the function? The function is sodium and silicon. How it happened? This is from here. How it happened? This is due to this one. How this has happened? This one, the master. And once the glomerular hydrostatic pressure increases, there will be starting of compromise. Lot of role of angiotensin 2 is there. The angiotensin 2 will cause the stimulation of aldosterone secretion, the stimulation of ADH secretion. ADH will cause the retention of water, and aldosterone will cause retention of the sodium. Once sodium and water remain, sodium will increase gradually the arterial blood pressure, and water will increase gradually the fluid volume. That's what we are talking. That means anything is decreasing, increasing in the body is sensed by those beautiful sensors. And that makes the juxtaglomerular apparatus a structure with a dynamic function. So with this, we came to the conclusion of this particular lecture. Hopefully, this lecture was very interesting, but the most interesting part of this is coming in the next lecture. That's known as the renin, angiotensin, aldosterone system. So stay tuned for my next lecture. Keep learning. Thank you so much. Keep learning. Thank you.